All right, let's dive right into a topic that's absolutely essential for your MDCAT biology paper, the cell wall. Nailing the key concepts we're about to cover is going to be a game changer on exam day, so let's get straight to it. Here's our game plan. We'll kick things off with a basic definition, and then we're going to peel back the wall's 3 layers. After that, we hit the most important part, the chemical makeup, which is a favorite for examiners. We'll finish up by putting the wall and the membrane head to head to clear up any confusion and give you a final checklist of things you just have to know. Okay, section 1. What is a cell wall? Let's start with the absolute basics to build a strong foundation.
So in its simplest form, the cell wall is a non-living, rigid, and protective outer layer. Now, if there are 2 words you need to burn into your memory for your MCQs, they are non-living and rigid. Trust me, so many questions are designed just to check if you remember these 2 fundamental properties.
So, where do we actually find this structure? Well, it's a key feature in plant cells, fungi, bacteria, and algae. But here's the classic exam trap they love to set. It is completely 100% absent in animal cells. Knowing this single fact can be an easy point for you on the MCAT. Let's move on. Section 2 is all about the wall's 3 layers. We're going to do a quick structural breakdown. Take a look at this diagram. It shows you the structure from the outside in, which is a sequence you have to remember. We start with the outermost layer, the middle lamella. Then we move inwards to the primary wall, and finally the innermost secondary wall, which isn't in every cell. Okay, let's start with that middle lamella. This is actually the very first layer that gets made when a cell divides. The easiest way to think about it, it's basically cellular glue. It's made of a substance called pectin, and its main job is just to stick neighboring cells together to form tissues. Simple as that. Right just inside that glue layer, you've got the primary wall. The key thing to remember here is that it's thin and, most importantly, flexible. And why does that matter? Well, it's because a young cell needs to be able to grow and expand, and this flexible wall allows it to do just that. It's made mostly of cellulose. And finally, once a cell is all grown up and has stopped expanding, it builds the secondary wall inside the primary one. Now, this layer is the complete opposite of the primary wall. It is thick, it is rigid, and it is incredibly strong. That extra strength usually comes from a tough compound called lignin, which also helps with support and waterproofing. This comparison is just perfect for your exam revision. Look at the key differences. The secondary wall, it's thicker and rigid, it's all about strength. The primary wall, thinner and flexible, it's all about allowing for growth. And here's a big one. Lignin is present in the secondary wall for that extra toughness, but it's absent in the primary wall. These are classic points for MCQs. Okay, I need your full attention for section 3. We're about to cover the critical chemical compositions. Seriously, this is a high-yield exam trap where a lot of students unfortunately lose marks. Let's make sure you're not one of them. So the big question is, what are these walls actually made of? Well, the answer changes depending on which organism you're talking about, and that's exactly where the exam will try to trick you. This table right here is pure gold for the MDCAT. You have to memorize these pairings. Let's go through them. For plants, it's cellulose. For fungi, it's chitin. And for bacteria, it's peptidoglycan. Let's lock that in again. Plants, cellulose. Fungi, chitin. Bacteria, peptidoglycan. Do not get these mixed up. Let's just take a slightly closer look at that bacterial cell wall. The main component, peptidoglycan, sounds complicated, but it's really just a unique polymer. It's made of these 2 alternating sugars, you'll see them called NAG and NAM, that are then linked together. This complex structure is totally unique to bacteria, which is why it's often the target for antibiotics. All right, on to section 4. Let's do a quick showdown, wall versus membrane. We need to clear up this common point of confusion once and for all. Let's break down the key differences, head to head. The wall is non-living and rigid. The membrane, on the other hand, is living and flexible. The wall is fully permeable. Think of it like a net that lets almost anything pass through. But the membrane is selectively permeable, acting more like a bouncer at a club. And never forget their positions. The wall is always on the outside. And that brings us to our final section, your MCAT checklist. Think of these as the golden rules you absolutely must take with you into the exam hall. All right, let's do a super quick, rapid-fire recap for your MCQs. Number 1, the cell wall is non-living and rigid. 2, plants, they use cellulose. 3, fungi, they use chitin. 4, bacteria, they use peptidoglycan. And 5, the very first layer that's formed is the middle lamella, and it's made of pectin. Lock these 5 facts in your brain. So after this focus breakdown, really, there's only one question left. When you see a cell wall MCQ on your exam, are you ready? After walking through these key concepts and those common traps, you absolutely should be. You've got this.